deciding which structure you want to use. But there is other information about X-ray crystallography that you should understand when comparing structures, so you can tell which structure is most suitable for the analysis you want to do. Now, X-ray crystallography here, I'm just going to give you a brief introduction. You have some protein in solution which you precipitate out, but it needs to be precipitated out in a regular pattern. And so you add some precipitate, high salt, or a volume exclusion agent like PEG, and that'll precipitate your protein out in a regular order. It's not a random aggregation. You want a particular order of the molecule stacking to make that crystal. Those crystals are exposed to X-rays and you get a diffraction pattern. So the X-rays interact with the electrons in your atoms and they scatter. And they're going to scatter according to the pattern of the electrons or the packing of the molecules within your crystal. Now I'm going to just wave my hands here, although this is a real time sink for the experimentalist. But part of the information the experimentalist gets is location of the spots on the film and their light or darkness. That's half the information they need to solve the structure. The other half is they need phases. And there are different techniques for getting those. I'm not going to talk about those. But once they have the amplitude and the phases, the amplitudes sort of tell you they're going 30 miles an hour or 40 miles an hour, but not telling you the direction, whether they're going south or north or east. But the phases give you direction. Once you have both pieces of information, then you can calculate the electron density map. That electron density map is interpreted by the experimentalist. And they fit atoms into that electron density map, and you get an atomic resolution model that you have here. That is your structure. And it is this structure that's deposited into the protein data bank. Now, what the experimentalist does is they take this structure and they will calculate a predicted diffraction pattern. And they compare that predicted diffraction pattern to the experimental pattern, and that gives them a sense of residual error. How well does my structure fit the diffraction pattern? How much of the data am I fitting? And the error is what's left over. How much am I not fitting? So they minimize the difference between the predicted diffraction pattern and the experimental diffraction pattern during refinement. And then when they reach a plateau, they evaluate, is this reasonable for this resolution structure? And then when they're finalized, they submit the structure to the protein data bank. The information that you see here are two pieces that are critical. Well, you don't need to worry about unit cell lengths or angles or that. What you do need to look at is the resolution and the R values and the R frees. Those are the residual errors. Now, resolution, as you can see on this slide, is sort of counterintuitive. And that is the smaller the number for resolution, the higher the quality of the data. And so you'll see here the electron density around this particular ligand at 2.85 is kind of blobby. By the time we get to 1.80, you start seeing more definition. You start seeing almost individual atoms. By the time you're at 1.1 angstrom resolution, you can actually see the whole in this tryptophan ring. And so the higher the resolution, the higher quality structure you're going to have, and the better the fit of the structure to your data. Now, in your lower resolution structures, around 2.5 to 3 angstroms, you're not going to see hydrogens in the structure because they're too small to be seen. But as you start going to higher resolution structures, less than 2 angstroms, down around 1 angstrom, you start seeing the individual hydrogen atoms in your structure. Now, the reason a smaller number means a higher quality structure is because that number reflects the spacing between your measurements or your data taking. So the closer together your planes are, the more data points you have and the more information. It's equivalent to smaller pixels. And so you get a higher quality image of the structure itself. So if you're comparing structures, most of the time you want to go to the higher resolution structure if everything else is equivalent. A more indirect way of looking at the quality of the structure is the residual factor, the R value and the R free. Now residuals are comparing the difference between your experimental data, your diffraction pattern, and the predicted diffraction pattern from your structure. The R value reflects the data that you use to refine or determine your structure. And the R-free reflects the data that is not used to refine the structure. And so what the experimentalist does at the beginning of the experiment, they will randomly pull out 5% of the data, and they will set it aside. And they will not use that, they will not target that in the minimization procedure. So the 95% of the working data is what is targeted in the minimization, in the refinement. And so the experimentalist, because it's a very complex pattern going from small changes in the structure to overall changes in your diffraction pattern, sometimes you can make changes here that are fitting noise in your minimization procedure. What will happen then is if you make changes here and your R value lowers but your R free stays the same or goes up, the experimentalist should reject those changes and try and do a new fitting. So that R free acts as an independent assessment about the quality of the refinement. Now, back in the mid-'80s, this technique came in, and if you find structures from that period, you'll see about 10% difference between an R value and an R free. You're likely to see less than 5% difference between the R value and the R free today, because we have better refinement techniques, better fitting of the structures to the experimental data. But generally what you're looking for, if you're comparing two structures, is you want to take the data with the lowest R value. Now a typical R value, R free, can be about 20% residual error. So there's still a lot of information in the data that we're not able to fit with the experimental models that we have. So those help you compare two structures and say, what's the better quality structure? One important thing you need to look at when you're pulling down X-ray structures is the asymmetric and biological unit. Now we're almost done with the lecture part of this workshop, and we're going to soon get into running PyMOL. But this is an important part that I want you to understand, is when you go and download structures, you're downloading the asymmetric unit. Most of the time what you're interested in is the biological unit. And the question is, what's the difference between the asymmetric unit and the biological unit? Now the biological unit is the oligomeric state of the molecule. It's in solution, in vitro. So I want to know what that molecule looks like in my test tube, in an aqueous solution. That's the biological unit. Now your asymmetric unit is what is the repeating unit in the crystal. So there is a pattern in that crystal. And the question becomes, what is the repeating unit in the crystal? Because the asymmetric unit is the smallest repeating unit in the crystal. What's my unique unit that repeats? So it's the smallest repeating unit. Now you can quickly visualize the asymmetric unit and the biological unit by clicking on these arrows. You can toggle back and forth for 1 dBt. Now when you're downloading the PDB file, you can just go to rcsb.org. You can click on download, and you have these options. If you just download the PDB file, either text or compressed, gzipped, you're downloading the asymmetric unit. And sometimes, maybe even a lot of times, it's okay to be downloading the asymmetric unit, because that asymmetric unit can be as big as the biological unit or bigger. But there are some times that your asymmetric unit is smaller than the biological unit. Let's say it's just a monomer of a homodimer because the twofold of the homodimer lines up to a crystallographic twofold. Then you actually only have half of your biological unit. In that case, if you want the full biological unit, you can come and download here biological assembly 1 or 2. Now, the reason I do not recommend doing that automatically by default is sometimes when the protein data bank makes these biological units, they don't change the chain name, and it'll confuse programs like PyMOL. So if you run into a situation where you want the biological unit and you can't figure out what's going on, and you can't make your browser or your viewer, your molecular viewer, show you the whole molecule, then you need to come find me and I'll help you with that process. But if you can download the asymmetric unit and get everything you need, that's preferred. But in some cases, you need to download the biological unit, and then we need to go in and edit the file in order to get it to work. One way you can look at the biological unit is the chain names that are being put up here are chain names for the asymmetric unit. And so you can just look right here, and this is an example where we have a single polymer, one polymer type chain, but we have 3 copies of that molecule. So we have a homotrimer in our asymmetric unit. So it's 3 chains, 1 protein. This particular structure is an example of 3 chains, 3 proteins. So we have polymer 1 is a G alpha subunit, polymer 2 is a G beta subunit, polymer 3 is a G gamma subunit. So this is a G protein, alpha, beta, gamma, but this is a heterotrimer. So the earlier example was a homotrimer. This is a heterotrimer. So each time we have chain A, chain B, and chain G. Now, the experimentalist gets to name the chain whatever they want. Most of the time, it's easy to figure out why they named it that. Sometimes it's not. But the chain is just sort of random. Now, what I want you to do is I want you to pull up these 4 PDB files. And I want you just to look at how many chains are in there. And so you're going to tell me how many chains are in the asymmetric unit. Now, these are all aromatidine decarboxylase structures. I'm going to tell you now that the biological unit is always a dimer in these 4 cases. One LOQ, how many chains for polymer 1? 1. So that's just a monomer. 1 LOL, how many chains? 2. For 1 DBT, 3. And for 1 DQX, 4. So these are all the same molecule. They all function as a homodimer. But because they come from different species, they crystallize with slightly different packing. And so the relationship between the asymmetric unit and the biological unit is different for all of these structures. Now, the first case, 1 LOQ, is the one case where if you're in a hurry, you're going to make a mistake. And I say this having done this, even after 10, 15 years working with structures. If you get in a hurry and just pull down the PDB file, you're going to look at this, and there's not going to be any clue that you don't have the whole molecule there, because you have the crystallographic twofold lines up against the molecular twofold, so you have just half the biological unit. but it's not obvious because your ligand is sitting in the middle of the molecule, right? So there's no visual cue there that you have only half of the molecule. So in the cases where your biological unit is larger than your asymmetric unit, those are going to be the cases where you may not realize I don't have the whole molecule. Now, in all the other cases, you're safe if you pull down the asymmetric unit, because for one LOL with XMP bound, the asymmetric unit and the biological unit are exactly the same. And not only can you see that you have a homodimer here, but you can actually see there are contacts between the ligand that's majorly bound to the green monomer, also makes some contacts to the cyan monomer. And so you need to be careful, for example, if you are analyzing contacts to ligands, that you're working in the context of the biological unit. But in this case, the asymmetric unit is the biological unit. For one DBT, you have one biological unit here, the green and cyan, and 1/2 of a biological unit, the magenta. And so if you want to be working with the 2 biological units, you need to go pull up the other half of the magenta one. Most of the time, I will just delete the magenta and analyze the green and cyan because they tend to be very similar. Yes?
So the only way to really know, make a determination, is actually look at the structure, compare they give you the number of colors.
Right. So the question is, how do you know, is it by comparing the number of polymer chains with the list that they're getting? In this particular case, the example you have here is you have a big visual cue, and that is the green and cyan chains have a lot of contacts, and you have very little or maybe even no contacts to the magenta chain. The other case you can look at to determine what the biological unit is, many times, not always, but it's listed in the PDB file. And so we're going to be looking at a PDB file shortly, so you can display the header to the PDB file. And that's going to tell you, in many cases, what the biological unit is, because it's provided by the experimentalist. If for whatever reason they don't report it, the protein data bank can run some analyses and try and guess what the most likely biological unit is. But in this case, you have 1.5 biological units, and in 1DQX you have 2 biological units. Now these are actually far apart, and so it's clear that you have 2 biological units here. And the reason they can be far apart is due to symmetry. If you actually go in and put up all the molecules in the crystal, you'll see that there's actually molecules making contacts all over the place that you're not seeing here. But the experimentalist has just picked 2 molecules which fulfill the symmetry, but they're working with whole molecules, and they just end up being far apart when they actually build the structure. And so that's one thing that you really need to be very careful about when you are working with X-ray structures. Now this is true only of X-ray structures because that has to do with how the structure is solved. So you can look in the PDB file, and it should give you some information about the biological unit. Most of the time it will, not all of the time.